Carol Ann and David Flanagan Professor in the Coulter Department of Biomedical Engineering at Georgia Tech Emory University. She is also currently the Director of the NSF Engineering Research Center in Cell Manufacturing Technologies and the Director of the Marcus Center for Therapeutic Cell Characterization and Manufacturing. She is interested in scaling culture of therapeutic cells and tailoring the molecular interactions between glycosaminoglycans protein cells for use in regenerative medicine applications. Her laboratory focuses primarily on promoting repair after injuries to the tissues of the shoulder, including cartilage, tendon, and muscle. She has a long list of prestigious awards, including NSF Career Award, Arthritis Foundation Investigator Award, SFP Clemson Award, College of the Fellows of the American Institute for Medical and Biological Engineering. She is also a Fellow of the Biomedical Engineering Society, a Fellow of the International Academy of Medical and Biological Engineering, and a Fellow of the Biomaterial Science and Engineering. She has also co-authored a very highly successful introductory textbook, Biomaterials, the Intersection of Biology and Material Science by Temenoff and Mikos, for which Dr. Temenoff and Dr. Mikos were awarded the American Society for Engineering Education's Miriam Wiley Distinguished Author Award for Best New Engineering Textbook. The floor is yours.
Thank you. Thank you so much for that very kind introduction. And I'm very happy to be here this afternoon. So I wanted to kind of give you an overview of what our lab has been working on for the past several years in terms of rotator cuff muscle. But I'd like to start out just wearing the hat of my director of our Engineering Research Center on Cell Manufacturing Technologies and tell you a little bit about that and how that's kind of informed directions in our laboratory over the past several years. So to jump in, this Engineering Research Center, or CMAT, the Center for Cell Manufacturing Technologies, was formed officially in 2017. And it was very fortuitous since this center came into being at approximately the same time as the first cell therapies were approved by the FDA for cancer treatment. So these are the CAR T therapies that many of you are familiar with. And kind of to show you the background and the mission of the center, I think this popular press article from the time kind of highlights these issues with these newly evolving cell-based therapies. And so, while these have the potential to be curative therapies and have been for a number of both adults and children with various leukemia and lymphomas, the issue really is the price tag associated with this. And this has to do with both the manufacture as well as required hospital stays due to side effects from many of these therapies. So we really believe that in order to improve access to these therapies, we need to ultimately reduce costs. And we think we can do that through bringing engineering to bear on this, including more efficient manufacturing of these cells. And so that's kind of the crux of what our center was developed to address. So in the process of leading up to proposing this center to NSF, we actually brought together in a number of listening sessions various key stakeholders in the field. And this includes clinicians, people in academia and industry, as well as government agencies, including regulatory agencies. And through a series of workshops, we collectively defined grand challenges in cell manufacturing. And this is eventually was created into a roadmap that we now have publicly available. But just to kind of run through a few of these, what we really just determined that was one of the major impediments to moving the field forward was this lack of reproducibility in many of our cell-based therapies. And a lot of this has to do with we don't really know what quality attributes define a potent cell for most applications. So it's difficult to pick out good cells from a highly heterogeneous population. Along with that, we can't actually then measure critical quality attributes or metrics of quality in line and non-destructively to feed back in and actually improve our culture as it's going through the process. And that then prevents us from growing the large numbers of cells that we would need to treat a very large patient population and make sure that they're all of high quality. And then other issues that are associated with all of the first four here are then how to predict safety and potency for particular indications and to better understand what patients may better respond to a certain therapy. And then if we can kind of answer all those questions, we then run into all of the now somewhat common supply chain issues that we've all run into with the recent pandemic. But all of this is very much the same for cell-based therapies: how to purify, store, freeze, and transport them and make sure that they maintain potency. So what we're really trying to do broadly within the field, but specifically with the research that we're doing in the center, is to get to end-to-end manufacturing of cell-based therapies at low cost and high quality. The other thing that we heard over and over again from our listening sessions was that there's really a lack of trained cell manufacturing workforce. So even those people that are coming from manufacturing other biologics, such as monoclonal antibodies, the processes that we have to go through to culture cells at large scale are sufficiently different that they need to be at least partially retrained. So this is a very unique product because anything that we do to it along the way in the manufacturing process can affect the overall quality of the product. And so we really need to have a set of people at all levels, all the way from technician level to PhD level scientists that are running the R&D arms, that really are intimately familiar with the challenges of manufacturing cell-based therapeutics. So this, as mentioned, ended up as sort of a roadmap that we took very much to heart as we were developing the mission and vision for our center. So the vision for CMAT is to transform the manufacture of cell-based therapeutics into large-scale, reproducible, and a high-quality engineered process, which we hope to do through, of course, developing new knowledge and new technologies, but also addressing some of those workforce and education issues. So the center itself is headed at Georgia Tech, but it does have a number of key partners, both within Georgia, as well as we partner with the University of Wisconsin-Madison, as well as the University of Puerto Rico-Mayagüez as our major partners. And then we have affiliates throughout the US and the world. And there are a number of faculty at each of these locations, and they each bring unique aspects towards the research that we're doing. The other kind of unique aspect of an engineering research center that's actually mandated by NSF is our industry partners program. And so here, I need to update this slide. We now have over 40 industry partners. And what we're very excited about is that these partners not only span all different sizes, so we have everything from startups to very large companies here, but they also span the value chain of cell production. So there are a number of companies that are providing raw materials. There are others that are actually producing the cells that are going to be eventually going to patients, and everything in between. So we have a lot of different perspectives at the table. And this group provides us with obviously the ability to easily transfer some of our technologies and hopefully smooth the path to commercialization. But also this group of companies reviews our research projects every 6 months to assure that we are meeting the needs of the field as a whole, and that there's not some new thing that's come along, because this field is really evolving very, very rapidly, that we may need to be aware of in our research. So this really allows us to meet the needs of industry as they're trying to define what the next products should be for the field. So another way to look at this is that we take input from these advisory boards, including industry, including our standards organizations with which we're partnered, and that then feeds into our research projects, which we have now kind of divided into 3 categories or thrusts. The first is what to measure to ensure cell quality. The next is how and when to measure it, particularly developing technologies that are inline and non-destructive. And then finally, how to scale to get to the number of cells that we really need. And we have technologies and projects that fall in each of these thrusts. And then they're applied to one or more of what we call our test beds, which are different cell types. And so the test bed here is therapeutic immune cells, so T cells and NK cells, and this comes directly from those FDA approved products. We also have mesenchymal stromal cells, which I'll be talking about since that's an interest in my lab. This was chosen because there are a lot of clinical trials, but not yet an FDA-approved product. And then a little bit more forward-looking are a number of iPS-derived cells that are just starting into clinical trials. And what we're trying to do here is make sure that our platform technologies are applicable across different types of cells that we might want to manufacture in the future. And so if we take all of this as our research, the output then is, of course, we hope new innovations and tools, and some of those examples are shown here. But also what we hope to do with our partnerships with standard organizations is help to transfer our knowledge quickly so that new standards can be employed and any information we learn, particularly about potency of cells, can be incorporated into an approval process at some point. And then finally, we have a lot of efforts in workforce development, as I mentioned, to kind of address those needs as well. I won't have time to go over any of that today, but we do have a very strong both K through 12 outreach programs, as well as courses and other training programs for undergraduate and graduate students. So now I would like to jump in and talk a little bit more about the work that my laboratory is doing underneath the umbrella of CMAT. And so, as I mentioned before, we have a longstanding interest in mesenchymal stromal cells. And while there are a couple different mechanisms of action, what we're focused on in the center and in my lab is MSCs as a source of secretory factors that are known to be pro-regenerative, anti-inflammatory, angiogenic, all of these sorts of things. And this is largely the mechanism of action of many of the clinical trials. So now this is a little bit of an outdated number, but there are well over 1,400 clinical trials that have been logged using MSCs of some sort. And you can see here that there's just a wide variety of potential applications, from neurological to musculoskeletal to cardiovascular. So there are a couple of issues with these. As I mentioned, there's actually no FDA-approved product despite all of these clinical trials. And we think that there are a couple of main issues in which the technologies of CMAT could be brought to bear. So one of these is that the typical dose in any of these clinical trials is often hundreds of millions of cells per patient. So if we were to look at something that has a very large patient population, like osteoarthritis or cardiovascular disease, we don't have the capacity currently in the US to make the number of cells that would be needed to treat this large patient population. So scaling is a big issue. And of course, we're very interested in those sorts of technologies in CMAT. The other issue that has led to, thus far, as I mentioned, no approved product, is that there's variable clinical outcomes for these trials. And we think that that is because there is a variety of cell sources, as well as different ways that these cells have been cultured prior to going into these clinical trials. And so standards and, again, understanding quality could be very important in just understanding and getting the best results from a particular clinical trial. So this is why this cell type was chosen as one of our targets within CMAT. And as we got together as researchers, we kind of identified a number of concerns having to do with both quality and cost that we are going to try to address within the center. And in terms of quality, I just mentioned that there's this variable success in the clinic. The other potential problem is that as we expand these cells to get to the number of cells that we need, this can cause a progressive decrease in potency and ultimately senescence of these cells. So we, of course, would like to avoid that. In terms of cost and risk, most of the concerns generally center on the need for biologics for expansion, which can be both costly and there's regulatory issues. So we have organized ourselves into a number of projects across the center that address each of these concerns. And so in the interest of time today, I'm going to be focusing on one of the ones that we're undertaking in our lab that have to do with conditions for expansion with reduced senescence and improved secretory capacity for MSCs. So the way that we've approached this in our lab as a biomaterials lab is using a hydrogel type of biomaterial based on polyethylene glycol diacrylate that we've used for many years in the lab. And what we are able to do is through various formulations, we're able to achieve gels that are about 30 kilopascal in stiffness and about 100 kilopascal, which you might hear me refer to as soft and stiff gels. And what we're trying to do with these is make alternates to the tissue culture plastic cell culture standard culture platform. And so what we've done is kind of made a family of these gels that are that include different cell adhesive peptides and different glycosaminoglycan derivatives, because that's an interest of my lab. So various heparin derivatives as well as hyaluronic acid. Included these covalently, made these hydrogels. And what we're interested in doing is actually seeding cells on top of these gels as an alternate tissue culture surface. So the results from this study show that there are certainly distinct morphological differences between the 30 and 100 kilopascal gels. Also, when we look at cell number after 4 days, the 30 kilopascal gels don't show an increase in cell number. This is our TCP control, our tissue culture polystyrene control. However, on the stiffer gels, the 100 kilopascal gels, we do see cell numbers that are similar to TCP over 4 days, suggesting that there's more proliferation on the stiffer gels than the softer gels. Interestingly, when we push this a bit further and we do 3 consecutive passages on our stiffer gels, in this case containing RGD, the cell adhesive peptide RGD, versus on tissue culture polystyrene, we can see that if we stain for senescence using beta-galactosidase staining, we can see that over 3 additional passages on each of these surfaces that we see an increase in senescence on the TCP, but we actually don't see that on our gels, suggesting that there's something very interesting going on here with this particular stiffness that we can see still maintain proliferation, but have a reduction in senescence. So this is very exciting to us, and we're continuing to try to understand more at a molecular level what is going on these gels. Going on with this experiment, looking at just one passage, we also wanted to look at what the cells were secreting as a measure of potency. So again, seeding cells on either the tissue culture plastic or hydrogel, culturing them for 4 days, taking the supernatant, running a multiplex ELISA, and then looking at multivariate analysis to help us understand broadly what the cells are secreting on each of these surfaces. So here, the easiest way is to probably look at the heat map. And here we have a heat map of all of our different surfaces. And again, similar to what we saw with the proliferation, the stiffness seems to dominate over any sort of particular ligand that's been added. And we see broadly with red being upregulated that there's an upregulation of many of the cytokines that we are testing on our softer surfaces compared to either the stiffer hydrogels or the TCP control. So we did a number of checks, but of kind of bioactivity of what was secreted. This is just one example. So in this case, we wanted to look at the ability of the supernatant, or what is secreted from the cells, to induce angiogenesis. We saw that there was an upregulation in VEGF secretion, for example, on the 30 kilopascal surfaces. So here we did a standard HUVEC in matrigel endothelial tube formation assay. We placed the conditioned media from the cells on top of the various gels here for 24 hours, and then looked at endothelial tube formation and the length of those tubes. And what we see is kind of across the board, again, regardless of the type of gel that the cells were cultured on, if they were cultured on 30 kilopascal surfaces, they induced longer endothelial tube formation than that on TCP. That was much more prominent in the 30 than the 100 kilopascal, and that's kind of shown here in the images below as well. So we certainly have the indication here that the culturing on them on these softer surfaces does improve angiogenesis, which can be very important for a wide variety of regenerative activity. So just as a brief summary of this study, we can see that we have both proliferation and what the cells secrete is sensitive to substrate stiffness on these hydrogels, and that we see that alterations in angiogenesis potential parallels the secretome changes. So while this was very interesting, this is where we were very interested to get the feedback of our industry members. And what they really said to us was, well, this is a very promising technology. What you have here is an alternate to culturing on planar surfaces like tea flasks. And again, going back to this idea of scale, if we really want to get to the numbers of cells that we need, which is, hundreds of billions of cells per lot or more, and we want to get that cost down to something that's commercially viable, we need to move from tea flask production to something like a suspension bioreactor. So this kind of shows you that, this is the scale that we can get to, and this is kind of the price point that an optimized culture might get to. And so they really encouraged us to move into a technology that would be more amenable to suspension bioreactors. And for adhesive cells like MSCs, that means that we need to have microcarriers that the cells could attach to and expand on. And so, however, when we started to look into this, a lot of the same questions applied, in that there hasn't been a lot of work done on how microcarrier stiffness or substrate biochemistry affects what the cells, how they proliferate and what they're secreting. So we kind of set up to do a similar study, but in a microcarrier fashion for our next set of studies. And so this is a recent study that is just now being published. But basically, one of my former graduate students took the formulations that we had previously used that we're calling 30 and 100 kilopascal, and used a microfluidic device in our laboratory to produce microcarriers with these materials. And of course, we can tune the size of these microcarriers. And this was important because we wanted to match a control. In this case, we call it Synthemax. This is a trade name for a tissue culture polystyrene-based bead that is used very often for MSC scale-up in suspension bioreactors. So we wanted all of these to have a similar size, which is somewhere around 200 microns in diameter. So you could easily compare between the three different surfaces. So the other part of this study was to kind of expand the cell types we were using a bit. And I should mention that in all of these studies with the MSCs, we use at least 2 donors and we see similar trends. I'm generally only showing you the data from one donor just to be clean, but we do see confirmation of trends across donors. However, these were all unmodified MSCs. And so what we, of course, are noticing particularly as CAR T therapies become more and more accepted, are that we have the need for, or the potential for using various types of genetically modified cells, including MSCs, that might overproduce a particular therapeutic protein of interest. So as a proof of concept here, we wanted to compare the response of a genetically modified MSC compared to a standard MSC on these various carriers. So in this particular case, we used an AAV transfection that you can see we were able to get these cells to overexpress IL-1RA, which is in our hands. We just chose this because a collaborator had interest in this protein as potentially reducing the effects of osteoarthritis, which we're interested in. But broadly, we can think of this as just a proof of concept overexpression vector for a potential therapeutic protein. So the overall hypothesis here was, okay, we know that some of these materials may increase the level of endogenously secreted proteins. We saw that on the planar surfaces. Would this have an effect on something that's already modified to overproduce a particular protein? So that's kind of the underlying question here. And so the experimental setup was very similar. We have MSCs or our transduced MSCs that are allowed to adhere to our various types of carriers for about 24 hours. And then we agitate them for about 3 days in well plates. So this is a small scale, but it does kind of mimic what we would see in a larger scale bioreactor. And then collect both the media and cells and look at the proliferation and look at what's been secreted into the media during that time. We also, in parallel, did some PCR to look at gene expression of key genes, and I'll come back to that in a little bit. So when we look at cell number over time, again, after 4 days, we do see some similar trends as we saw in the planar surfaces, in that the Synthemax, so the TCP-based material, and the stiffer microcarrier have a similar cell number, and there's less cells available or present on the 30 kilopascal, the softer gels. As you'll see for all of these data, the modified versus unmodified MSCs behave very similarly. So when we look at secretion, first of all, when we look at the transgene encoded protein, we can see that there is an elevation of secretion of the IL-1RA on the softest substrate compared to at least the Synthemax TCP control. However, when we look at the endogenous secreted factors, there's a difference from what we saw on planar surfaces. So instead of seeing a broad upregulation of factors in the softer hydrogel microcarrier, we are seeing specific factors that seem to be secreted at higher levels, including hepatocyte growth factor and IL-10. So while we don't see this broad upregulation, we are still very interested in this because these are known to be pro-angiogenic anti-inflammatory factors. So they might still have a potential to be very pro-regenerative in vivo. But there is obviously a marked difference here. And we're trying to continue to understand a little bit more about what's different between the carriers and the surfaces that might cause some of these differences in secretion. So this is just to show you one snippet of showing both donors here. And this is the secretome very broadly, but what you can see here is that, each of these surfaces, each color represents a different surface, does have a unique secretome, and that largely the modified and unmodified cells in every case of being the stars and the circles do overlap. So again, we're not seeing a large effect of the genetic modification on the secretome overall. So given the fact that we saw upregulation of very specific proteins on our 30 kilopascal microcarriers, we went ahead and probed a little bit more for gene expression on two of these, the overexpressed IL-1RA and hepatocyte growth factor. And interestingly, we saw as early as day 1, so prior to agitation, that we saw an increase in gene expression of both of these genes on the softer microcarriers. And so this suggests to us that whatever changes are occurring that result ultimately in protein secretion are happening very early as the cells are adhering on these gels. So we're continuing to probe into this and try to understand what signaling pathways might be involved in something that's going on here. But we thought it was very interesting that this is happening very early on for these cells. And then finally, for completeness, we wanted to take that supernatant, just like we had done with the planar surfaces, and look at this HUVEC assay again to see if there was bioactivity of what had been secreted. And again, similarly, we do see an increase in endothelial tube formation on the softer microcarriers as compared to the other types of substrates. And we think this is largely due to the increase in secretion of HGF, which is known to be a pro-angiogenic growth factor. So just to show you a little bit about where we're going with this then, my current graduate student, Keshav Shah, is taking these microcarriers and actually thinking about ways that they might be able to be used in vivo as well as in vitro. So originally, in the first iteration of these, within the context of CMAT, we are interested in making them as alternates for microcarriers that might be used in bioreactors, and therefore did not really add in any sort of degradability. However, when we're thinking about transferring this into our in vivo system and eventually using cells to do things like help repair rotator cuff injuries, of course, we would want a material, a potential delivery material to be degradable. So what he's done here is actually converted our 30 kilopascal formulation to one that includes an MMP degradable peptide, and been able to show that we can, again, tune the diameter of these microparticles with our microfluidic device, and that we are able to put these in collagenase, and we see complete degradation of these materials over 1 day. As a first proof of concept here, and this is just with one donor so far, we went ahead and did some of the very same studies. So seeded cells and looked at cell number over 4 days. And we can see here that actually there is no difference between our microcarriers and the standard Synthemax microcarriers in terms of total cell number. However, when we do this multiplex ELISA for what is being secreted after 4 days, we see a very similar situation in that there is very particularly HGF and IL-10 that is upregulated on our degradable microcarriers. So this is very similar to the non-degradable carriers. So whatever is going on here seems to be reproducible and has perhaps more to do with the stiffness than the actual biochemical composition of the material. So just to summarize where we are with this set of studies, we have found that we can make hydrogels into microcarriers with these various stiffnesses. They've been made as degradable or non-degradable versions. With our study, we can show that there's an increase in transgene encoded protein secretion on these 30 kilopascal microcarriers versus our Synthemax. And there's several other factors that are differentially regulated. However, there is some difference between what we see on the microcarrier surfaces, particularly on the 30 kilopascal, versus on those planar culture surfaces. And we're still trying to understand a bit more about what's driving those differences. All right. So I wanted to kind of switch gears now and talk about other aspects that are going on in the lab that are outside of CMAT. So the way I think about our lab is that we are divided into kind of two strategies that can come together to help regenerate musculoskeletal tissue injuries. And so the one that I have been talking about, I call it more like an exogenous strategy. And this is where you're going to take cells, autologous or allogenic cells and culture them separately and then reintroduce them into the defect. And that is all of the work that, of course, we are trying to do through CMAT and eventually move back into in vivo situations. The other side of the coin, though, for some patients, maybe that, this strategy is not necessary. And that we might be able to actually recruit the body's own stem cells or other pro-regenerative cells to help jumpstart this regenerative process. So I call this more of an endogenous process. And what we're trying to do is also design therapies that would recruit these endogenous cells using biomaterial and protein delivery strategies. So with that, I had mentioned previously that our lab has a long history of being interested in rotator cuff injury. And there's just as a brief background, I'm sure that many of you know someone or yourself are suffering from a rotator cuff tear. It's very common pathology, and it increases with age. And so this is well known. What's less well known is kind of how this happens over time. And so what's generally thought is that there's some sort of tendinopathy that starts at some point caused by repetitive motion. It ends up with a disorganized ECM, as you see here. So the tendon should be very well aligned, and at some point it becomes less aligned. And there can be pain. There can be, perhaps even not even that much pain. So it's not clear that everyone knows that this is going on, but it predisposes the area to rupture. And so at some point, there's a tear of the tendon. It can be partial thickness or full thickness. And there's actually 4 different rotator cuff tendons and associated muscles that are found in your shoulder joint. And so these tears can be involving 1 or more tendons. And this is where you typically lose a lot of function and you have severe pain. So our laboratory has several different animal models. The one that I'm going to talk about today is that a model about severe rotator cuff injury or massive rotator cuff injury. And this is a tear that involves more than one tendon. And so what happens here is that when you have this tear, you unload the associated muscle, and this leads to muscle atrophy. And so when we look at it, an MR, and this is something that one of our clinical collaborators has provided, here we have the humeral head, here we have the clavicle, and then this area right here is the muscle, and it should basically not be white. So all of the white is some sort of degenerative scar, if you will, which is usually composed of both fibrous and fatty tissue. And the issue here is that, first of all, that this happens, because of the unloading. But unlike other muscles in your body, when the surgeon goes in and reattaches the tendon, this just adding load back onto this muscle does not reverse the atrophy that's occurred. So a lot of patients will have significant loss of motion even after their tendon has been reattached. The other issue with this atrophy of the muscle is that the muscle pulls back towards the midline. And so the surgeon has to pull the muscle tendon unit very tightly to get it all the way back to the humeral head to do the suture, as I've kind of shown here. And so this causes a lot of tension at the suture area and actually leads to high re-tear rates. So for both of these reasons, we would like to think about a strategy that actually targets the muscle that could be used in conjunction with current surgical techniques that would allow reattachment of the tendon. And this thus far has been an understudied area of rotator cuff injury, is kind of what happens to the muscle and how do we either reverse atrophy or at least prevent atrophy from getting really serious. So for this, as I mentioned, we have a rat model of rotator cuff tear, and we actually transect two tendons, and we inject various of our strategies, of our therapeutics into usually the supraspinatus muscle directly here for these studies. And then we have a contralateral control that is uninjured for comparison. So I'm just going to kind of share with you one vignette about some of our most recent work in this area. And so just to give you some background, what we're interested in doing is in part delivering local factors that might recruit these endogenous cells. And one of these is SDF1 alpha. And so this will actually attract a number of CXCR4 positive cells, including the ones that are listed here: lymphocytes, leukocytes, mesenchymal stem cells. So this gets back to kind of the first half of the talk with our interest in MSCs, as well as a number of other progenitor cells. So this was some of our first work in this area in conjunction with Ed Bochwe, in which we placed soluble SDF in one of our hydrogels and placed it in essence, a subcutaneous model in a mouse that had it's a chimeric mouse that has all bone marrow derived cells fluorescent green in this mouse. And what you can see here is that underneath the gel, we were able to recruit a number of bone marrow derived cells when the gel was releasing SDF, but we did not see this localization when we just had a blank gel. So this really showed us that we could have an in vivo strategy that would indeed recruit cells through the vasculature and into a localized area. So with that information, we actually then turned to making microparticles that were more easily injectable that might carry this SDF. So to do this, we actually used a GAG derivative. This is a heparin derivative that has been desulfated so that it still has a negative charge, so it can interact with the positively charged stromal-derived factor 1, and has a hydrolytically degradable component as well. And the main reason that we picked this particular formulation is that this formulation has, because of the desulfation level, does not have the anticoagulant properties, so we don't have to worry about a lot of bleeding, and it's very helpful for any sort of surgery. So we still have the ability to load and release in a controlled manner the SDF without some of the negative side effects that you might associate with heparin. So we made these microparticles And then we did a variety of studies just to show you here that we were able to inject these into the supraspinatus muscle and track these over several days and show that with tagged SDF that the tagged SDF persisted longer locally when we had the microparticles than just a bolus injection of SDF. So the results from these studies then also showed that given this, that we were able to attract more MSCs into the supraspinatus muscle when we had the treatment than when we just had blank microparticles. So that was all kind of the background of the next set of studies that we then wanted to ask. So we, had shown that we had locally injected microparticles and that were able to attract some, at least a certain number of cells from the vasculature. So what we wanted to then talk about was something called a push-pull hypothesis. And so the idea here is that you can attract a certain number of cells if you inject locally and release SDF locally. However, what happens if we combine this with a systemic bone marrow mobilizing agent so that you would push these pro-regenerative cells into circulation and then pull them to the site of injury with that local SDF release? And so with this study, we wanted to use the same injury model and wanted to look at, the systemic delivery of a bone marrow mobilizing agent and at the same time as an intramuscular delivery of these SDF1 releasing microparticles. And the idea was that we would be able to see more pro-regenerative cells in the what we call the dual treatment than either of the treatments alone. So part of this, just to let you know, was to pair our SDF strategy with the proper bone marrow mobilizing agent. So most of the clinically used bone marrow mobilizing agents target CXCR4, which is also what we're targeting with SDF. So that would render the SDF portion somewhat useless. So we had to find a different mobilizing agent, again, with the help of the Bochwe lab, that uses an alternate strategy for mobilization from the bone marrow into the blood. So with that, we can use this, what we're just going to call the mobilizing agent, but it uses an S1P gradient to get cells into the blood that then can still be responsive to SDF1 alpha. So to go through the experimental setup here, we basically had 4 different groups, what we're going to call dual treatment, each of the treatments alone, so mobilizing agent alone, the SDF1 alpha alone, and then an injury for comparison. We looked at all of these groups up through 7 days, and then we actually took only the dual treatment and injury groups on for days 14 and 21, looking at both flow cytometry to see what cells were present, as well as histological markers of muscle degeneration and regeneration. So when we look at macrophage phenotypes, and this we're going to focus on largely what's happening in the muscle at days 3 and 7, we can see that we have a larger number of M2-like macrophages used with the mobilization agents. But when we do the M2 to M1 ratio, and this is basically pro-regenerative or pro-resolving macrophages to potentially more pro-inflammatory macrophages, we can see that in both cases, the dual treatment and the mobilization agent have a higher of this sort of ratio at day 3, and this persists with the mobilization or the dual treatment only up through day 7. But when we look then at further time points, there's really not a lot of difference. So the differences that we see between groups seem to largely occur within the first week. And I should mention that the injury was done at the same time as injection of these therapies. So this is a very acute treatment for an acute inflammatory environment. But we do seem to be able to alter that type of inflammatory environment based on what we're delivering here. And then when we look at MSC recruitment, when we look at MSC mobilization into the blood, we actually see as early as 24 hours with either the dual treatment or SDF, we're getting mobilization of MSCs in the blood. And then at day 3, both dual treatment and mobilization agent, you see more MSCs in the injured muscle. And again, this persists with the dual treatment up through day 7. But again, not a lot of change beyond that. So again, we're looking at kind of some acute changes in the cellular milieu. And then Finally, we wanted to look at muscle stem cells and just a marker for T cells. And in this case, we looked at day 7, the PAC7 positive satellite cells, and we see those are actually up in the SDF only group. And again, not tons of changes over time. So we seem to be focusing more on the recruitment of the MSCs and perhaps changing the macrophage a bit more than impacting at least the number of satellite cells in the muscle. However, interestingly, by day 7, the dual treatment does show an increase in T cells as well. So there does seem to be, certainly, again, within that first week window, effects on both innate and acquired immune cells. And then finally, just to show some of our muscle fiber regeneration and morphology metrics. So in other words, we can change the cellular milieu. Does that have any impact on any of our regeneration metrics? So there are 2 here. One is embryonic myosin heavy chain, which is an early marker for muscle regeneration. And interestingly, here at day 7, we can see that the mobilization, the dual treatment is actually the only treatment that shows more of these positive regenerating fibers than SDF alone and injury. But the other treatments do better than injury alone. So we do seem to have some effects, again, on very early muscle regeneration with that dual treatment strategy. Centrally located nuclei are another measure of regenerating muscle fibers, and these typically happen in time. This metric comes on after the EMHC staining. So by week 3, again, we see dual treatment being higher than injury. So this kind of follows that trend. So this is all very interesting, and again, suggests that we are able to change things in that first week that does have some impact on the potential for regeneration in this environment. However, when we look at a little bit longer time points for fibrous infiltration and muscle weight, the overall conclusions are that we have not significantly affected levels of fibrous infiltration, and the muscle weight does decrease so that by day 21, you do get levels of atrophy that are similar between the treated and untreated groups. The issue here is that we actually did not do a reattachment in any of these animals. This was just an acute injury, and to kind of see what if there was a potential effect of this dual treatment. So what we believe is happening is that while we are able to alter early metrics of regeneration, that ultimately the environment is degenerative enough that ultimately we still end up with atrophy in these muscles. So again, the conclusions here are that, we are certainly able to change or alter the cellular milieu in terms of macrophage ratio and MSCs. We do see early markers of regeneration, although we do still see atrophy of these muscles over time. So we're interested in doing additional studies, perhaps with reattachment or other rehabilitation strategies that might be used in combination with these sorts of treatments. So with that, I just want to conclude by saying, of course, that we are very anxious to, continue to pursue both of these strategies. I don't think that there's necessarily one exogenous versus endogenous that's better. I think it may be very patient specific, but there are certainly things that we learn from both arms of the lab that can help us really just understand how best to treat, particularly muscle degeneration in a variety of patient populations. So with that, I would like to thank everyone in the lab, particularly those highlighted in red that did a lot of the work that I presented today, our funding sources and our collaborators, and I would be happy to take any questions.
Thank you for the fantastic talk, Professor Temenoff. Several questions came in. One is people were asking earlier on about the percentage or concentration of materials in the gel. You did the 2D. I think the question came in when you were showing the surfaces 2D.
Yeah, so there's actually a different weight percentage for the two different stiffnesses. I don't know it off the top of my head, but I'm happy to get it for you. And it's also in the paper.
Thank you. The second question is they were asking about how long are the microcarriers stable in media?
So we haven't done that, we haven't tested it for unknown periods of time. We are currently actually doing bioreactor studies with these microcarriers. So we're talking about scaling them up, and we certainly can do cultures up to a week without seeing a lot of degradation. We just haven't gone past that at this point.
Thank you. For the dual treatment, what type of release models are you interested in?
In terms of like linear versus or what, is there an additional, sorry, I'm trying to find the question. So that, I mean, in terms of an ideal release model, if we're talking about kinetics,
like a sustained or fast or bolus.
Okay. Yeah, I mean, certainly we would want something that sort of eliminates as much as possible the burst release, because we would like this to be sustained over time so that we can continue to recruit cells. And so, does it necessarily, I guess, ideally, we always want it to be linear, but does it absolutely need to be linear? Probably not, but some sort of definitely sustained release. We were looking to target at least a week. And actually, I didn't show that data, but in our most recent studies, we're now able to take the sustained release in vivo out to about a week.
Thank you. What type of cross-linker do you use for PEG gels?
So the PEG-DA gels cross-link to themselves because they have acrylate groups, and we do add in different types of functionalities that can allow them to be degradable. So we have a DTT-based gel that is hydrolytically degradable. We have that MMP cleavable sequence that we can add in if we would like it to be enzymatically degradable. And then we use two different types of crosslinker. We use a UV light initiated crosslinker for some of our studies, and we use just a thermal radical initiator, APS and TEMED, for crosslinking in some of the other cases.
Thank you. Another question came from LinkedIn. Can you generalize your microcarrier delivery for other muscle diseases?
Yeah, so that's our hope. I mean, we are, that's one of the reasons why we're developing the collagenase sensitive carriers, because certainly MMPs are upregulated in a wide variety of muscle diseases. So this is something that we would like to do on the cell side, and on the protein delivery side, all of our microcarriers are hydrolytically degradable, which should be very compatible with muscles throughout the body.
Thank you. For the postdocs in the audience, what are remaining problems they can focus on moving forward in tissue engineering biomaterials area?
Yeah, so I think I mean there's still a lot of challenges. So I think it could go in one of two ways. One is this idea of how do we continue to push towards commercialization or towards adoption in the clinic, right? And so for that, we really need to think about making sure that you're doing reproducible work, which is a lot of the focus of CMAT, and making sure that you're really writing down and doing a clean design of experiments to really optimize things to get it very, very reproducible. So I think that's kind of for everybody, just as something to think about that I was probably less aware of when I was more junior, just how important it is to think through both the design as well as the execution of the experiments and show how reproducible your materials and techniques are. In terms of the, kind of the pie in the sky science, I still think that there's a lot to be done in terms of spatiotemporal release of either cells or growth factors, and really trying to match that with what is needed in various regeneration or degeneration models. I don't think we quite have enough find enough control over either of those aspects right now.
Well, thank you so much. That's all the questions I have. I appreciate your time, Professor Temenoff.
All right, it was great to be here today. Great to chat with you, and have a good afternoon.
Thank you. Bye-bye.